Hello, everyone. Welcome to Chapter 28, Part 1. So, in Chapter 28, we are going to be discussing the reproductive system. So, this lecture, we're going to focus up through the male reproductive system, and then we'll save the female reproductive system. Basically, we're going to do the first 40 slides of this chapter. Which really takes us through the male reproductive physiology, but this is a very large chapter. It's 95 slides, but I want to talk about that. So this is our very last lecture chapter. So I thought it fitting because reproductive system is the beginning of our story in life. So I thought every story has a beginning and an end. What lies between those 2 points is the journey. For you guys, this is the end of your anatomy and physiology journey. Most of you guys are going on to take some other courses now. Many of you are going to be applying to nursing school in the next semester. So as we go through this, let me help you guys understand it. Now, what I want to do is, as always, I like to iterate that for my courses, what I do is I always test on the chapter objectives that we lie out here. There will be something from each one of these. One of the things that I think works as a great system of study is, can you do this? Like, can you describe the hormonal changes that occur during pregnancy, explain the role of hormones in the birth process? Now, I have asked about that. It is not these things here, embryonic development and pregnancy, are not in this chapter. There are separate chapters on that, and they are not in our current syllabus. I have been asking about this. This needs to be revised. Number 10 and 11, I can tell you, are the only two that we don't do. It is actually not in the current chapter as it stands, not since I've been here. And I've been doing this I've been here for 10 years. So that means these objectives have not been updated enough. They go through updates, but they have only been updated one time, and that's not something that has been brought up. Even though I bring it up in multiple meetings, it's just not something that's been adjusted. So I'm going to say that we can't do it because there are no concept there in this chapter, and the chapters that do that are not on the current syllabus. And then I wouldn't be able to actually teach it because the syllabus says this is what I got to teach. Though I would like to, and I can. My first graduate level course was actually on reproductive physiology specifically. So that was my very first graduate level course I ever walked into. So definitely could do it. I had embryology. Like to do it. I have some embryology videos, some videos that discuss a little bit about fertilization and some of the basic embryonic development on my YouTube channel. So please feel free to watch that if you're watching it. So anyway, without further ado, let's get into this. Now, the first thing I want to talk about is kind of always with any system we deal with, we always have some functions that we talk about. Now, the functionalities of this is, well, it does a lot of things, but this is the only one I don't need to actually keep myself alive. If I lost the reproductive system out of a body, they're not going to die as long as, they have proper care after, as long as, but there would be alterations to their quality of life, but you need it to produce life. You need it to reproduce, to make babies. Now, this system really just is consisting of four major structures, and we're talking about the gonads, that'd be testes and ovaries, those ducts, things like the vas deferens and the oviduct that transport the gametes, accessory glands, things like the prostate or the greater vestibular glands or Bartholin's glands, the external genitalia, the penis, the vulva, things like that. Now, remember, we have gonads. Remember, gonads, these are the structures that produce sex hormones and also sex cells or gametes. The male gonad is the testes. The female gonad is the ovaries. The male gamete is the sperm. The female gamete's an ovum. Now, notice I say ovum and not egg. An egg is a fertilized ovum. And that's when we call it an egg, is upon fertilization. Now, one of the four big things we do, we can produce offspring. Okay, this is the ultimate thing. We produce offspring. Now, we also nourish offspring. So this is why we will discuss the breasts in female anatomy in this chapter as well, because they help nourish the offspring. The uterus, the placenta, these structures help to do that. Now, I wish there was more discussed about the placenta here. This chapter just does not do that. We discussed placenta during chapter 21 in that physiology. There's some things here that I'd like to discuss, things that you may deal with, like placenta previa, things like that you may deal with. Some other diseases I'm going to add. I'm going to be adding PCOS and some other stuff to these chapters. Some male stuff, prostate. At least probably 2 male and 1 more female disease. I got placenta previa in my current edition. But some of these structures, there are issues that happen with them, and it's very important that we discuss them because they lead to some of the issues that can lead to reproductive health. So we do nourish the offspring, breasts, placenta, things like that. Uterine lining for the first week. Your uterine lining, the glands that secrete, the endometrial glands actually nourish the early stage embryo in the first week of development prior to implantation. There's gametes are produced, sperm and egg. Those gametes get transported through things like a vas deferens or an oviduct or fallopian tube. And we also make our sex hormones, our testosterone, our progesterone, and our estrogens are all produced via the gonads. And the gonads produce a lot of these important sex hormones. Now, we're going to start with men, and let's just give a little bit. Now, I could tell you guys that in my exam, I have never really gone in and asked very specific things about the size, but I do want to give a general size of a testis. Now, these are really more for reference. Men's testes can vary in size and shape, but they're average. They're about 5 centimeters in length, 3 centimeters wide, and about 2.5 centimeters thick. So they are roughly kind of more like a bean, somewhat elongated, okay, and more like a bean. Kind of think about beans a little bit wider than they are thick. Quite a bit longer than they are wide, kind of a more like a bean shape. They weigh about 10 to 15 grams, and they are in the scrotum. Now, these are kind of average subsets of descriptions of these, and I do not test on them myself. What I do want to talk about, because there are things that happen, men can have enlargements or reduction in size, and that is an important thing through part of a normal male reproductive checkup. The size is often given. What the doctor will do is kind of a double thing. Oftentimes, like if you go to a doctor and they are checking that for males' reproductive health, they will have models and they will ask, they will compare it, they will feel, then they will ask the patient sometimes to feel their own testis and say what they think, which one feels the closest to how they feel. And if that is average, if the male has has too small gonads, this can affect some things. So just some basic information. Now, the scrotum is the pouch that encloses them. It's below the perineum, pelvic floor. It's actually in front of the anus, but behind the penis. So it's anterior to anus, posterior to the base of the penis. So it is in between the penis and the anus, hanging in the perineum area, the so-called pelvic floor, the perineum. Now, in males, there is a structure called a spermatic cord. Now, the spermatic cord, it passes through the inguinal canal, which is left because of male's testicular descent. And this is where, like, the ductus deferens will be located, the blood vessels and the nerves and all the lymphatic vessels that come to and from testis that are important. That is the beginning here. The spermatic cord starts at the inguinal canal and travels through into the scrotum. And it's this area that's between the abdominal pelvic cavity and the testes here as it descends downwards through. And inside here, that inguinal canal is a passageway, and inside that is a muscle called the cremaster. Now, Cremaster muscle is one of those muscles that helps us maintain a body temperature, a testicular temperature, temperatures of the testes that is about 2 degrees cooler than average body temperature. Remember, average body temperature is 98.6 degrees Fahrenheit, 37 degrees Celsius. We want to be 2 degrees cooler than 98.6 degrees Fahrenheit. Now, with Celsius, 2 degrees, I do not believe is the correct value. I'm going to hesitate to say because I did not write that down. And but 98.6 degrees Fahrenheit is the common thing that people use in America. But in science, we're going to use the Celsius. We're going to use scale. That to me makes far more sense. Base 10 scale, easy to make conversions on other units because you just move decimals. But growing up in America, if you say 37 degrees Celsius, now that's pretty hot. I mean, you imagine here, we know what 98 degrees feels like here in the South, in southern United States. We know what that feels like. We've experienced it, okay? And so that is pretty warm. That's our body temperature. Okay, so what I want to do is talk about the tunica vaginalis and dartos. That is in the scrotum. Now, the scrotum, I have a tunica vaginalis that acts as the serous membrane that lines the inner lining of scrotal cavity and lines the testis. And then that reduces friction as it produces a fluid because it is a serous membrane that transudate serous fluids can help reduce friction, which also can reduce damage, temperature production, things like that. Just make it far easier, less wear and tear on the testis. The dartos muscle is right under here in the skin. You have the dartos. The dartos fuse to the skin. This causes the testes to be wrinkled. And it also helps elevate the testes as well, along with the cremaster. Now, as well, if we look on the outside, we had what was called the tunica vaginalis, which you can actually see here and here. But under it is the tunica albuginea. And the tunica albuginea or albuginea is going to be the dense irregular connective tissues, and it has this tissue goes inwards, forming these walls called septa. And these septa help to divide the testes up into various lobules. And the lobules have about 800 centimeters of seminiferous tubules in them. That's roughly 32 inches. And these little seminiferous tubules produce sperm. Here you can actually see the seminiferous tubules. There are many of them, very small ducts, and they produce the sperm. And then what they will do is they will send these attach to this area here we refer to as an anastomosis, and this is called the rete testes. And the rete testes will go up to the epididymis using these tiny little small ducts called the efferent exiting ductals. And that takes us to the epididymis, where we can mature sperm. There's was about the head, the body, and tail of epididymis. The epididymis takes us to the vas deferens, and the vas deferens is where we can carry this to the ejaculatory duct. It ends at the ampulla. So this transports and stores sperm in the vas deferens. It ends at the ampulla. Now, the ampulla is best going to be seen right here on that diagram. Now, what I want to do is talk about, here you could see the seminiferous tubules. We could see sperm producing. They produce a lot, spermatocytes, and there are a lot of them. Now, the epididymis, there are 3 different parts of it. I thought I had one that did depict it. I need to add this image. I have an image that does have it. It's just that it's on a disk, and I need to go upload it into here. But we have the big part, the head, the body, and the tail. Head is really enlarged. This is where you're getting all the efferent ductals, the head of the epididymis, the body, and the tail. The tail is the inferior part that attaches to the ductus deferens or vas deferens. Sperm is stored down here. They really help to mature throughout the epididymis. One of the things we do is the fluid inside here gets monitored and adjusted. Anytime we have spermatocytes that get old, worn out, or damaged, we recycle them, but we also basically do what we can so they can be stored, protected, and they can grow up and mature to become good quality sperm. The chemistry of this environment needs to be conducive for sperm maturation, for the sperm to be good enough to be able to fertilize. Male reproductive health does help to kind of center around some of that. This is why, like, if a male has low testosterone, he may have trouble conceiving. Sperm don't tend to mature very well, things like that. If his testes remain too warm, stuff like that. So it's definitely rough. Now, as we talk about the glands that are using to make ejaculate, now a male produces about 2 to 5, like 2.5 to 5 mils, let's say about 2.5 to 5 milliliters of fluid during a normal ejaculation. And that fluid is made by 3 major glands. One of them that makes the most is called the seminal vesicles, seminal glands. The seminal vesicles, seminal glands are on the back of the bladder. Just here we have the ampulla of the vas deferens here. We have a seminal gland and its duct going in to form the ejaculatory duct. Now, that is, the ejaculatory duct receives the seminal fluid during an ejaculation. And then the prostate is the next gland. Notice that it surrounds the urethra, which means it can impinge the urethra upon urination, making it difficult. It secretes an antibiotic compound into the semen. The next two, which I'm going to zoom in so we can see better, you can see one here. There will be one on the other side called the bubourethral glands, sometimes called Cowper's glands. And if you kind of look at it, the way I remember they're called Cowpers, they have these 2 horns like cow horns, bull horns, steer horns. I know cows don't have horns, it's the bulls. You know, steers can have them because a steer is a castrated bull. But anyway, these little guys here, they make mucus to neutralize. This mucus is alkaline or slightly basic to neutralize any acids in the urine, because we know that urine contains acids as we play a role in excreting that out during renal compensation for an acidosis. Okay, now the vas deferens. As it comes down, we actually have the ampulla. As it enters, the ampulla enters into the ejaculatory duct, and it's bringing with it the sperm that mix with seminal gland secretions with the seminal vesicle duct, seminal duct to the seminal gland, and that mixes. And in here, there is one of the big components to this, and this produces by far the majority of ejaculate, is inside of it is fructose. And fructose, we know, is a monosaccharide. It is an isomer of glucose. It has the same chemical formula as glucose, just a different structure, but this becomes the food for a sperm cell. As I used to always say in class, is that sperm, they have a very long swim ahead of them, so they pack a lunch. They got a long journey, so they're going to bring their food with them. So it's like if you're going to be gone all day, you're like, well, I better pack a sandwich, and things like that. So I always say, they have a long swim, so they pack a lunch. That's fructose they take with them to feed the spermatozoa, so they have the energy to power their flagella to swim towards and fertilize. Now, in the male reproductive tract, there is a molecule called prostaglandins, and this stimulates through muscle contractions. Prostaglandins, we know, cause pain and inflammation, and these molecules can actually go and cause muscle contractions as well. In this, this is the thing. I'm not trying to be gross, but sometimes, as you guys deal in healthcare, you will be asked, but that molecule, among some other things, can, because these prostaglandins cause pain and inflammation. You can imagine part of this could be why that people will sometimes say, like, well, if they get, and I'm having a hard time, but saying this in a way, I'm trying to be kind of proper about it, and also a matter of fact clinical about it, is so you will get questions that are sometimes kind of uncomfortable. And somebody may ask you, well, why is it if they are engaged in a specific sexual act? Let's say that they're like, well, why does it hurt? Why do they get a sore throat when they swallow seminal fluid? Well, part of this is why. Now, you could just say chemicals in semen, okay, and it irritates the tissues. But you will be asked these kinds of questions. And I'm not trying, because these kind of get really embarrassing and difficult to address and really difficult to answer sometimes because of that. And I do want to help you with that a little bit, because I can tell you, you get asked it. You get asked it when you're in education, and you get asked it when you're in patient care, because one of the big jobs that especially nurses have is patient education. And you need to be able to answer these. And I'm going to tell you, you need to be able to answer this with extreme professionalism. A former student of mine, they were working in a hospital, and they had a male patient who they were working on, had an issue, and one of the nurses lost their job for complimenting the male patient on his anatomy. You cannot do things like this. Okay, so you have to be able to kind of handle with maturity these really uncomfortable things, and always be professional. Okay, it's so I'm trying to be very, like, I'm using this opportunity to kind of tell you things that I have seen and have gotten people, like, when you get asked these, you have to be just very clinically honest, brutally honest. I'm not trying to be gross, not trying to be disgusting, trying to be clinically honest as I absolutely can possibly be. Now, there are that alkaline stuff. It neutralizes the acid in the vagina as well, that are in seminal fluids, as well as the urine, like with the bulbourethral glands. Now, the prostate itself circles the urethra. What it's got, the antibiotic that it produces. Now, we'll talk about, but it's got smooth muscle in it, and it will squeeze and eject its secretions, and it's got seminal plasmin, the antibiotic that's in there that can help, especially in the female reproductive tract. Now, bulbourethral or Cowper's glands down here, they are going to make a thick mucus that neutralizes the pH of the urethra, but it also helps to lubricate the tip of the penis. This produces a so-called pre-ejaculate. Now, when someone uses the different terms that are sometimes used in common vernacular, I do correct them. I always use appropriate terms, clinical terms. Now, most of us will already leave public life knowing those words, because you hear them, the so-called words that are used, like, you probably learned them in middle school or earlier when people started talking about sex. But we will always call this ejaculate, and you guys know, but the pre-ejaculate is that. Now, there sometimes can be spermatozoa in the pre-ejaculate, depending upon how recently the male has ejaculated. If that number is sufficient, and it theoretically can be, it is possible that even though a male is having unprotected sex, he can theoretically get a woman pregnant. He can potentially get a woman pregnant even without ejaculating during unprotected sex. So, those are things. These are the kind of things you deal with. I will say that I have probably never dealt with more difficult questions than it was when it comes to reproductive system, where the questions always, to me, kind of lead me to a shock of the ignorance amongst the general population about their own bodies. Ignorance is not pejorative. I'm ignorant on a great many things. The human body is where my education lies. I am very ignorant on pop culture. I am very ignorant on a variety of things, and I recognize that ignorance. But I can tell you guys that if you understand, it also, I would say, get a knack with dealing with this in a very professional, somewhat. It takes tact. There is a very good way to deal with those things, and that is something you get with experience. You'll have to do it. Just remember, if you're, it's I would even sometimes say it's better to be golden clinical at first, but it's very hard to get the like, hey, it's okay. I mean, I always kind of approach it with, it's okay that you don't understand this. Let me kind of show you this is not something everybody knows. It's okay that he said, but this is important that you understand it, and just kind of say, hey, it's okay that you don't know it, but now that you do, please let me help you understand it. And that's the thing. Okay. Now, I want to talk about in semen. Like I said, it's around 2 to 5. I might say 2 to 5, 2.5 to 5, somewhere around there. Mils. There is about 20 to 100 million sperm per milliliter in semen on average. Normal. I have seen higher numbers to 200 million sperm count. And these are numbers that I'm using. So we're going to say 20 to 100 million per milliliter. Now, you think about this. Our book says, now I have read some different books, but 2 to 5 mils. I have read 2.5 to 5, a variety of things, but 2 to 5 mils. Let's just take that one. If you have 20 to 100 million sperm per milliliter, and there is, let's just say, 2 times 20, 40 million, or to 200 million sperm per milliliter. I mean, in that semen, okay, that's a lot. That is a huge number, and you could be 5 times that. Okay, imagine that number. That is a huge number. Spermatozoa for men. Now, if it's low, that can make it difficult for him to get a female pregnant. High sperm counts. There could be a few reasons for that. Part of this will be is how often the male ejaculates, his testosterone levels, the temperature of his testes, a variety of things of his reproductive health, age, stuff like that. It's oftentimes and very much so an odd thing is that we typically tend to find that when oftentimes that males are at their sexual peak is probably the worst time for them to be fathers, in terms of at least modern time, because just considering that they spend more time now at home, it's we're delaying our kind of adulthood, so to speak, in modern life in order to become more educated and having a kind of delaying our certain aspects of our life till we get other things. So seminal fluids, they are a mix of all the glandular secretions, the semen, from all your seminal glands, your prostate, the nerve cells of the testes, the epididymis, the urethral glands, everything that is coming out of the testes, everything that's coming out of the ejaculatory glands. And there are several enzymes that are produced by these glands, like proteases that help break down mucus. There's a protease, and this also comes in part of why sometimes you may find that if seminal fluid comes in contact with various mucous membranes in the body, it may become very irritating to people. And this is something in reproductive health people ask about. And it's sometimes it is kind of now with younger people a little bit more sexual liberated. It is kind of surprising sometimes they'd ask you that. But in a way, I'm very glad that they would ask. I would rather somebody ask me something and that before they really get themselves into some serious trouble with diseases or something else. So always kind of take that like the idea that when you were asked these uncomfortable questions, you want to kind of show that you're thankful they asked it, And it's to me, those are things I found personally that prevent people from being too afraid to ask. Is they say, okay, and you answer it honestly and you answer it openly. Seminal plasmin, the enzyme that kills the bacteria, that's from the prostate. There's enzymes that clot the semen, and there's fibrinolysin that actually liquefies clotted semen. This is something that will happen is upon ejaculation, semen will become thick and very almost clotted, and then it will liquefy. And this helps in it staying around just long enough for the other enzymes to do their job so the sperm cells can enter the vaginal mucus and be able to swim and be able to make it into the rest reproductive tract. So it's kind of needed. And then liquefies so this material may begin to drain out. It is it leads to one of the most uncomfortable questions I've ever been asked, which these things come in and rely on. These things help to answer as well. I'm not going to go into that right now. I have talked about it in classes a little bit because people have asked. So I've given a kind of a basic breakdown what these things do, what they may help, things like that. Now, erectile tissues, these help to produce an erection. An erection is caused by the parasympathetic nervous system. Ejaculation is caused by sympathetic. Parasympathetic causes, so you actually have, way I remember that parasympathetic causes erection is point, shoot, and score. Point, parasympathetic, causes erection. That's what makes the penis point. Shoot, ejaculation is sympathetic. Score, orgasm is sympathetic. And that helps you remember what neural aspects of that was controlled by which division of the nervous system. Point, shoot, score. There's corpus spongiosum, which is around the spongy urethra or the urethra, penile urethra, anyway. The corpora cavernosa, that's two pieces of erectile tissue here, and the spongy urethra is another name for the penile urethra. The glans penis is the head of the penis, which is covered in a prepuce. Prepuce is the so-called foreskin. That's what covers the head of the penis. We remove it during circumcision for those males that have been circumcised. Circumcision is a very common procedure done in the three Abrahamic monotheistic religions of the world. Those are common. Judaism, Islam, and Christianity, which are common that come from the Abrahamic traditions of the Middle East. Now, but not every religion in the world does this. And of course, depending on where you grow up and the kind of culture you grow up in, this is more or less common. And there are benefits, costs and benefits to both being uncircumcised and circumcised. It's a very serious thing to consider in your life if you're going to have discussions. Definitely recommend talking to someone who is an health professional about that. If you are considering about, like, if you're having a child, and that child you found out is male, is to really consider that. It is a very serious thing to discuss with your healthcare provider. There are costs and there are benefits. And I would never say for anybody that is something that is definitely a very difficult choice to make. Sometimes most people just do it because it's what they did. It's coming, from what I can tell, less common. But I do want you guys to, I'm going to say, hey, if you think about becoming a parent, take it seriously. Now, I use the mnemonic PMS to help remember the three divisions of the male's urethra because males, my anatomy professor, and I always kind of get a giggle out of it every time, and when it comes to reproductive system, we are dealing with some things that are very serious and somewhat embarrassing for a lot of people, which also means that sometimes just a sense of humor helps us kind of alleviate the tension of talking about a topic that can be, for some people, very difficult. And my anatomy professors always say that men have a wastewater treatment plant right in the middle of an amusement park. And what he meant is the urethra is part and parcel of two body systems. Wastewater treatment plants, the urinary tract. It's involved in the urinary tract, but it also is part of the reproductive tract because semen passes through the urethra in males. So males urinate and ejaculate out of the same structure. So I always remember that, and it's just something I always kind of gives me a little chuckle back to my days of human anatomy, and learned a tremendous amount from that professor. And some one of the things I use is PMS to think prostatic urethra, membranous urethra, spongy urethra, PMS. Now, the prostatic urethra is only the part of the urethra that you would find located here, but in the prostate itself, within the prostate. Membranous urethra is this very short portion that passes through the urogenital diaphragm. And then the spongy urethra or penile urethra is the part that goes in through the penis. Now, if we want to say PMP, but I prefer PMS here. So men do have PMS, prostatic, membranous, and spongy urethra. The opening here is called the external urethral orifice in the glans penis. Now, that opening here is used for both ejaculation, so ejaculate, seminal fluids, and urine pass through both of these. Okay. Now, a big thing that we're going to get into. So there is the anatomy. There is guys. We looked at their anatomy. I have found that generally, it depends on the group I'm teaching, but mostly men. I found one thing that men are always most interested to learn about is where their prostate is. And just real quick about prostate health, why is it that prostate? Men will sometimes put this off, and I'm going to do a kind of a PSA here. And I kind of find it funny because PSA, public safety announcement, but as we're dealing with a PSA about the prostate, I'm thinking also prostatic specific antigen, which is a thing that can help us check prostate health. So a PSA about PSA, the prostatic specific antigen is a thing that helps us tell maybe we might have an enlarged prostate. But there is a thing that is also that you're going to do. Men who are listening to this lecture, I'm going to tell you that if you go about your life and let's say you are using the bathroom, everything's normal, you're trying to urinate, you're not currently having an erection, which is common for men to wake up in the morning with nocturnal tumescence, having an erection when they wake up in the morning. And that's common. But if you're urinating and it's very difficult, you dribble, you have weak and interrupted stream, this could be sign of enlarged prostate. So when if this happens, now make a very specific doctor's appointment about it. If you feel like you really, if this continues on. This is one of those things, testicular cancer, prostate cancer, catch it early, catch it early, catch it early, and by all means, catch it early. You will hear me say that a lot when it comes about cancer is always the number one thing to ensure greater likelihood of survival is early detection. Now, the doctor does do what is called a digital exam. So their fingers will be placed inside your rectum. And why they do that is the prostate's right here in front of the anterior wall of the rectum. So when they push into that, they can feel that and locate and feel and see if it is enlarged to see if it's bigger than it should be. And this could mean impinging here. So there may be a blood test as well for prostatic specific antigen. This is a thing, depending on your age, you will start to have as a regular part of your examination. But if it happens that you are concerned, you can call your doctor. Don't try to do this. Is a very common thing men do. Is they sometimes go in, they have an issue. They'll say, "I'll just wait because I've got in 3 or 4 months, I've got a doctor's appointment. I got a doctor's appointment in 2 months. I'll deal with it then." In 2 months, a lot can happen. And then what you do is you go in. Your doctor is finally done, and a lot of doctors, they'll kind of go in, and as they're leaving, they'll sometimes, as they leave, they're like, should I say, because it looks like maybe their patient wants to say something, and sometimes they're not sure, and then they're hoping what they kind of call a doorknob question, and then they're like, doctor, sometimes I have a hard time peeing. Don't wait for that. Make an appointment specifically about it. Go to the doctor. And if a prostate exam is necessary, I understand you maybe have some trepidations about it. The uncomfortableness of what may happen, what is going to happen during that examination is going to be nothing compared to what you may have to endure with cancer. Okay, so please do that. Without those things, those are some things that I definitely kind of want to hit upon. Now, let's talk about this. This is something I talked about back in A&B 1. And there was a question I had in A&P 1, exam 1, where I said something like, if a cell has 46 chromosomes and undergoes mitosis or meiosis, how many chromosomes will the daughter cells have? Now, let's talk about this. Mitosis and meiosis, these are two important types of cell division. I talk about meiosis very briefly in A&P 1, and I tell my students you will see this when we get to A&P 2. Now, mitosis, this makes somatic cells. What are somatic cells? Remember, these are cells that are not gametes. These are body cells, neurons, muscle cells, liver cells, skin cells, that kind of stuff. Anything that's not a sperm or an egg. Now, one of the big things I want to help you guys understand here is it always makes exact copies. Always makes exact copies. So when I go to make my exact copies, what I'm going to do is I'm going to copy my DNA exactly. And I always make 2 copies of each chromosome. That fancy word for that is diploid. At least in humans, our somatic cells are diploid. We have diploid somatic cells. Now, you probably heard this. People talk about this all the time. It's one of the common things. You will see that there's a human diploid cell that is used. I've heard people say they use aborted fetal cells to make vaccines or in the vaccines. They're not in the vaccine. The cells that were used were there was an aborted fetus that was discarded. The cells were obtained. Those cells now have been kept under cell culture. They've been passed. It's been a long time since they were taken from that fetus, but you had to use real cells in the production of this to do that. They're not actually in the vaccination. They are a lung cell that is used. One of the reasons they're used, they are lung cancer cells. And they are used for that reason because you can take cancer cells and keep them in liquid cell flasks. Okay, those are easy to maintain and use in the production of those vaccines because you need to have something to test them on. For example, the MMR, sorry, not NMR. I'm not thinking about nuclear magnetic resonance. The MMR, mumps, measles, and rubella, which I have had myself, those vaccinations. And a lot of people talk about that, but those are diploid cells. Okay. Anyway, sorry. I was taking that chance because I was just having that discussion not too long ago with somebody. I do a lot of science education on the side. Okay. So if we have 46 chromosomes in a normal human diploid cell and it goes through mitosis, it will have 46 chromosomes in the daughter cells. Identical. Now, meiosis makes sex cells or gametes. Now, these are never identical. You never make this same sperm twice. Now, I mean, the sheer probabilities of that are so unlikely. A male probably never produces the same sperm twice. Just on the fact of, there's a huge number. It's 2 to the 46 power for a male. How many possible sperm he can produce that's without genetic crossing over. Pretty impressive numbers. Now, there are also only one copy of each chromosome. We call that haploid, remember. Haploid means you only have one copy of each chromosome. Now, people say sometimes half the chromosome number. No, you're always correct to say one copy of each chromosome. Okay? And why that is, it's half the diploid number. And one copy of each chromosome. Now, if somebody has, for example, you have those, well, half of 4 is 2, but that's still not haploid. One copy of each chromosome. Now, I would say half the diploid number. I'll count that. But I always say, if you want to be accurate, one copy of each chromosome. So if I have a cell that starts, its diploid number is 46, and it undergoes meiosis, the daughter cells only have 23 chromosomes in it. We have them. Always gets halved. Okay. Now let's talk about this a little bit. Let's talk about this process called meiosis. And there are a few things that I want to talk about. Meiosis is a two-part process. Okay, I'm so angry now. I forgot that this was unplugged. And now it's not going to work. That's what you get when I don't think ahead. I didn't plan ahead, and this may not work. Okay, good. It's going to work. And I don't know why. Sometimes it just does the weirdest things. Okay. Now, what I want to talk about is meiosis. And I'm going to do a few things because I'm running low on paper, and I don't want to use my rocket book anymore. I just kind of had to for purposes, but I'm going to split this in half and talk about two different things here. Now, what we want to do, let's talk about meiosis a little bit. Meiosis happens in two distinctive phases, okay? So let's say I've got a cell here, and this cell that I have has two copies of each chromosome. Now, when we do this two copies of each chromosome, what is very important here is that when I do this, when I have a chromosome in my body, here we have a chromosome that my mother gave me. Okay. And we have a chromosome my father gave me. We have maternal and paternal chromosomes inside this cell in its nucleus. And we would say this cell here is 2N or diploid. Okay, diploid. This means you are in fact two copies of each chromosome. Now, what's going to happen is, now we talked about this a little bit in A and B1, but we undergo what is called meiosis 1. Now, what happens here in meiosis 1 is a very interesting thing. And I'm going to draw these two cells, the products of meiosis 1, these two cells. But what I'm going to do is I'm going to put the red chromosome over here and the blue chromosome over here. Now, if you've had General Biology 1, this is Mendel's second law, the law of independent assortment that we see here. Okay, allele pairs segregate or assort independently. Okay, so what happens, there's a 50-50 chance, and it is basically a coin flip here. This is the first democracy right here. So, all right, now these cells, if you look at them, they are just N, haploid. They only have one copy of each chromosome. Okay, haploid. That was meiosis 1. Makes two haploid cells with one of the chromosomes in it. Now, these are called homologous pairs. I'm going to talk about that in a minute. Now, another thing that I want to do is, let's go. We got to do this again. Meiosis is so nice, we get to do it twice. And let's say that I undergo this process here. What do we do here? Well, we are left with these things called sister chromatids separate. And this was called meiosis 2, and these cells here are 2N, or just N rather, haploid. Now, one thing that happens in meiosis 1 is we say homologous pairs separate. What do I mean by homologous pair? These are two chromosomes of the same number. We have 46 total chromosomes in our body, 23 pairs. I have 2 of chromosome 1, 2 of chromosome 5, 2 of chromosome 10, 2 of chromosome 22, 2 of chromosome 23, sex chromosome. Okay? I have 2 pairs of every single chromosome. I have a pair of every chromosome. I have 2 of each chromosome, so 46 total. I do this for each one. I get one from mom, one from dad. I split these up. Now, this is one of the first reasons why you don't look just like your parents. Okay, when they make gametes, you don't look just like one of them, is because this is the first reason right here. This is actually a 50-50 split here, which is pretty cool. And then we separate this thing in meiosis II, and I say sister chromatids separate. Now, one of the things I like to make a point about is that homologous pairs, the red one and the blue one, are different colors. They're different colors because red and blue are different. They're different colors because red came from mom, blue came from dad, and mom and dad are different. If I got my red chromosome from my mother, my blue chromosome from my father, my mother and my father are not the same person, so we end up having two genetically different chromosomes. But the two red ones are red because they are genetically the same. They're called sister chromatids. These two reds are identical. They are the same. They have the exact same. They are the products of DNA replication. These two blues are the same, but red and blue are not because they are from different individuals, different people. So what we get is 4 gametes made. Okay, 4 cells are made by meiosis. Now, what I want to talk about is that also there is something here called that a cell goes through DNA replication. It makes, after it's replicated, you get these chromosomes that have 2 chromatids each. And they are 2 genetically identical chromatids called sister chromatids. You have one from your mom, one from your dad. Now, what happens with these, it's really interesting during meiosis, and we're going to take this, is they will line up. Now, I'm going to draw it a little bit different now, because for me, it's a little bit easier to do it this way, the way I'm going to draw it. And let's say I have a chromosome, and right here we have a centromere, okay? That's our centromere. The centromere is the center part where the 2 sister chromatids, 2 genetically identical chromatids, are attached to each other. Now, these are identical. Let me kind of explain this, that they are identical, okay? If you've had general biology, and also if you have me for MP1, we know that everybody's DNA is a little bit different. Now, the GT C's and A's on this red line is the same as the GC C's and A's in this red line. And the GT C's and A on this blue line are the same as that one on that blue line. But these two here are not the same. Why? This came from mom. This came from dad. And mom and dad are not the same person. Now, what is very interesting, if you had General Biology 1, then you know we have something called alleles. Now, let's say I've got a gene called A, and I'm going to put the one allele here, the dominant allele here. And let's say we have a gene called B here, and a gene called C here. Then this person here has A, but they got the recessive allele. Maybe over here, they got the dominant allele. And then maybe over here, they got the same allele. Okay? These two are identical. These two may not be. Now, you can see these two are not the same here. And here, they are the same here. Now, what they will do is each of these are genes, and they will line up gene by gene. Let's say they line up gene by gene. Now, what's going to happen, which is kind of interesting, we actually, when they line up gene by gene, lined up. We actually call this process synapsis. Sorry, I misspelled synapsis. Where they line up gene by gene. Okay? Now, if we count chromatids, these are chromatids, DNA strands. There is 1, 2, 3, 4 chromatids. Since there's 4 chromatids, we call this a tetrad. Okay? Now, synapsis, they line up gene by gene. What may happen during this is a process called crossing over. Now, genetic crossover is one of the most important processes of gamete production to take. Now, right here, this is the first reason why you don't look just like your parents. Even though you may favor one of your parents more, you don't look as an identical copy of one of your parents. This is another reason why, because we're going to basically even make it more different. Now, let me show you. If I undergo meiosis 1, homologous pairs separate. So these homologous pairs, I found that it's hard to say homologous pairs while I'm drawing at the same time. They separate. Now, let's say right here, I've still got those big A alleles. Remember, an allele is just an alternate version of a gene. Hopefully, you guys have seen that before. And then, okay, my little b right here is still here, but guess what happened? Right here, I hit that crossing over. Now, this didn't make any difference. It didn't change it in any way. But maybe it changed some other stuff that might be in here, okay? And I could have done a little bit more of a better example, but, it's okay. It's not like I haven't done anything bad. It's just like, okay, I probably could have done it a little bit better way, I think. But I'm always looking like, if I was thinking if I didn't have the same alleles there. But I also know you guys may not know what alleles are yet because it's not something we talk about in biology or in A&P that much. And you know what I was supposed to do here, and I'm having one of those. It's a day. I'm gonna blame it on it's a day. It's a day, guys. Okay, so and then what we're gonna do is exchange bits of each other with us, and then if we undergo meiosis 2. What you're going to end up with is these little interesting bits like this. And this can be extremely fascinating, okay? Because what you can end up with is sometimes some new variations that were not there before, okay? And that in itself gets very interesting. And you get these right here that are pieces that are mixes of mama and daddy, okay? Of inside my mother and my father. So if this was my chromosomes and they were lined up during synapsis, during meiosis, and they undergo crossing over, they can exchange bits of each other's genetic material between the two homologous pairs and end up with these two here that also have a bit of each of my parents in it. And if this is a sperm that fertilizes an egg, it can end up with a unique combination. And you get what is this then goes on to create genetic diversity. And it's a very important thing called a crossing over. Now, all 23 pairs can do this. They all do synapsis. They line up gene by gene. Very cool thing. They form tetrads during synapsis. During synapsis, we have a tetrad. Genetic crossing over will form, and it will break the genetic linking between those two. Now, meiosis 1, remember that you go through the identical chromosomes called the homologous pairs. They are non-identical homologous pairs. Now, what I want to do here, let's kind of label this. I'm going to use purple to label it because that, do I have the purple that I like? Okay, there we go. My favorite purple here. And we call these homologous, I cannot spell, sorry, homologous chromosomes. Okay, and we separate these homologous chromosomes, homologous pairs. And then here, let's use a color. I'm going to use orange, just so it's easy to tell. These are called sister chromatids. sisters identical, homologs not. I will never forget my genetics professor yelling at us in class. This is sister chromatids identical, homologs not. As he was Russian, and he would scream in his Russian accent to us. So, Dr. Yampolsky, if you're listening, I've never forgotten. So I will say this is PMAT twice. So let me help you guys understand it. PMAT twice. Now, what we do, and I'm not going to go through and draw this out. Really want to make sure you guys kind of, during prophase 1, like with mitosis, our chromosomes condense, and we have 2 chromatids for each chromosome. One is maternal, one is paternal. The red one, let's say the red one was from mama. Let's say the blue one was from daddy. And they gave me that. They are matched together. They will go and line up what we call gene by gene, called a tetrad. That is called synapsis. They literally line up gene by gene. And it's called synapsis. We have 4 chromatids who are in a set called a tetrad. Now, remember, the 2 red ones are identical. The 2 blue ones are identical, but the blue and the red are not identical. That's why I use different colors. Okay? Crossing over increases genetic variation. Now, one of the cool things is you never know which way the maternal and paternal chromosome will go during prophase 1. Which way after meiosis 1 anyway. Now, metaphase 1, those tetrads line up. Now, it's the tetrads line up on that, but they split randomly. They distribute randomly. 50-50 split. The meiosis was the first democracy. It's the first equal 50-50, 1 chromosome, 1 vote. It is the first democracy. That's how I always think of it. Now, anaphase 1. Maternal and paternal chromosomes as homologous pairs, they separate. One cell gets a maternal one cell gets a paternal one, and this is random. It's a coin flip. Once the coin is flipped, if one side is heads, the other side is tails, and then the fate is sealed. Okay? Now, then telophase, PMAT twice, we undergo our division. We have already made a haploid cell, the meiosis 1. It's already reductional division. It is already reductional. You've already split it in half. Because the 2 cells are now first to be haploid, the 23 chromosomes, they are now reductional. There is a brief interphase after this, reductional division at telophase 1. But you don't replicate DNA. We don't want to do that. We would undo the work we did if we did it, but you can't anyway, because you actually replicate chromatids, not whole replicated chromosomes. Now, meiosis 2 is just basically like mitosis. They're all the same. The chromatids separate during anaphase, and this is equational. Why is it equational? Okay. Here, if I had 46 chromosomes in a cell here I would have 23. And here I would have 23. This is why I say this is reductional and this is equational. Okay? Okay? Does it make sense? I hope it does. So you can't ask me a question. It's not like, part of me is like, I was just waiting for one of you guys to say yes, but I'm talking to no one here right now until you actually listen to it. So, all right. Now, very important bits here to do this, okay? And you can see what I've drawn here, meiosis 1, meiosis 2, all that good stuff, okay? They just don't show crossing over, and I think crossing over is extremely important to talk about. Now, let's talk about males and how they make sperm cells. And this is where we're going to go into meiosis, and we're going to talk about meiosis. I'm going to help you understand meiosis, and you're going to be like, oh, meiosis makes sense. What we're going to do is we're going to talk about spermatogenesis. And spermatogenesis is a process that men do. They start spermatogenesis. So what I'm going to do, I'm going to talk about gametogenesis. Let me rewrite that. Gametogenesis. Now, in gametogenesis, we will see spermatogenesis, and then later on, next class, we're going to see oogenesis. Okay? Now, spermatogenesis. Let's go in and focus on that. Now, in men, begins at puberty. Okay, that means that men, when we do spermatogenesis, we don't start making sperm cells until puberty hits. Okay, when men hit puberty, they will start to produce sperm, and that's it. Now, up in the testes, up in the seminiferous tubules, there will be a cell in here, and this cell is a diploid 2N cell. Let's prove it. 2 copies of each chromosome. I'm only showing 1 pair, but there's 22 others to go. So we got whatever we do here, guys, just remember you do it 22 other times, is a diploid cell, 2N, diploid, 2 copies of each chromosome. And this is called a primordial germ cell. Now, what I want to do with primordial germ cell is we're actually going to undergo a mitosis first. Now, when this cell undergoes mitosis, there'll be 2 cells. One cell replaces the old one because mitosis always makes genetically identical cells. And this will be a diploid cell, and it's going to be called a spermatogonium. Spermatogonial cell, spermatogonium. And it's going to be diploid, so we got to prove it. Let's make sure I prove it that it's, in fact, diploid, 2 copies of each chromosome. And then what's going to happen to it is it's going to undergo one more mitosis, one more mitotic division. Mitosis is used in male gamete production, So we're going to produce 2 cells here. Now, it shouldn't surprise you that these cells here will be 2N diploid cells, and they're going to be called primary spermatocytes. Okay, now we got to make sure we prove it. Okay. Okay, these cells have been made. All right, they've undergone mitosis. They are now there, ready to go. Now, we got 2 of these little suckers, so what we're going to do with these 2 suckers, let's follow one of them. The primary spermatocyte, what's going to happen to it? We're going to undergo meiosis 1. Now, remember, in meiosis 1, what you do, guys, is homologous pairs separate. And I always draw it like this. Why? Okay. It's because it can happen, so I'm going to have it that way. And we now have a haploid cell called a secondary spermatocyte. Okay, secondary spermatocyte. Now what we got to do with secondary spermatocytes is undergo meiosis 2. And we just write meiosis 2 here. And then we're going to get 1, 2, 3, 4 cells here. That's just from the one I'm following. You're also doing it here, so you can imagine. Okay. Where you're just going to get 1 chromatid now per chromosome. And you've got your haploid spermatids. And then what happens to these guys is they undergo a process called spermiogenesis. And that's when these little cells here will grow and mature to become little spermies. little spermatozoa, little sperm cells, okay? And what they basically do is they carry one copy of each chromosome in them, and that's your mature sperm. It's called spermiogenesis. Now, one of the big things I want to talk about is that's what we see here, or spermatozoa. Okay. And I am somebody who says, hey, if you can draw this out and sketch it like that, it's really not that hard to get down. Now, I can tell you one of my favorite questions to ask on this. Here's one. I'm going to ask a question. I'm going to see if you guys can answer it. I'll pause. When I ask it, pause before and I'll give the answer very shortly after. But when I ask this question, pause and see if you can answer it, and then come back. So, what is the first cell to be haploid? Okay, so if you said that it was a secondary spermatocyte, then you're right. This is the first cell that actually underwent a reductional division. Remember, secondary to spermatid is equational. Okay. So, we're going to come back to this definitely. I will use this to get the other half for oogenesis. But definitely an important thing to learn there. Okay. And I drew it out, and I really would rather just draw it than just say it. But I say it's a straight line meiosis. Why? Because it starts at puberty, goes through life. There's no pauses, there's no breaks, there's no stops. Men do this their entire life. Okay, so here you can see this. They start with a primary spermatocyte, go to a secondary spermatocyte, and things like that in spermiogenesis. I did the whole shmagegi. Now, the nerve cells who were involved in this, what do? The nerve cells are so important in making sure spermatogenesis happens right, that they're so crucial at making sure it happens right, that without them it doesn't happen right. And so what do? Well, one of the things they do is maintain a blood-testis barrier. Not a blood-brain barrier, but a blood-testis barrier. That keeps your immune system cells from coming in and destroying these cells, because these cells could be unique and your immune system could attack them. We also help hormones that come into this, especially FSH and testosterone. They're going to be helping stimulate nerve cells to cause spermatocytes to divide and help it. So these cells guide mitosis and meiosis. They're almost like sperm wranglers. They're going to kind of help to make sure that the mitosis meiosis happens properly. They also support the maturation of sperm, called spermiogenesis. That when a sperm cell's mature, they let them go and let them swim on their own. They make a hormone called inhibin when there's enough sperm around. When there's enough sperm, they release this inhibin. Inhibin goes to the anterior pituitary and says, stop releasing FSH. It's a negative feedback control mechanism of FSH release, which means the testes will stop making sperm. So if there's more sperm, there's more inhibin. And if there's more inhibin, there's less sperm production. And this will make the sperm production go up and down to make sure that we're only producing the sperm we need. Men, now that depends on how often the male is ejaculating, things like that. Also, the nerve cells produce a hormone called androgen-binding protein, which helps grab up the androgens to turn that into testosterone. Also, a hormone called Mullerian inhibiting factor, which causes your testing descent. It also causes regression of the Mullerian ducts. Anti-Mullerian hormone or Mullerian inhibiting factor, things like that. These things are what causes one of the major distinctivenesses between male and female reproductive development. Okay, so this is what causes the scrotum to descend, and males' testes may not descend if they don't produce this. They have undescended testes. Part of what happens in any male who's like, say, in elementary school, and you're going to go try sports, they're going to have a test they're going to do. And that's what they're testing for, is undescended testes. This may be something you have to do under a sports physical. Doctor probably also tests it. They usually check the abdomen near where the inguinal canal is to make sure they didn't get impinged there. Now, if they don't descend and they're still up inside the up inside the abdomen, that might be because of not producing malaria inhibiting factor. So when nurse cells here, when they help a cell here undergo and grow, they will grow and develop called spermiogenesis. The cell sheds, it goes out and now can swim and be on its own as a mature spermatozoa. And spermatozoa basically are these little nuclear torpedoes, basically. Why do I say nuclear? They have a big nucleus. The nuclear payload is the DNA. The chromosomes are inside this nucleus, the 23 chromosomes that this is carrying, one of which is either an X or a Y, and then one copy of chromosome 1 through 22. And so there's basically the first 22 chromosomes, and then the one, either an X or a Y. That's it, either an X chromosome or a Y chromosome. Males determine the biological sex of the baby. And this head in front of it has an acrosome. And it's somewhat like a shape charge, like a modern anti-tank weapon. We figured this out way back a while ago. For example, shape charges we use. You think of things like Piat round, Panzerschrecks, bazookas, Panzerfausts, things, the anti-tank weapons of the Second World War. You think about those kinds of weaponry. Still using shape charges to this day to penetrate armor. The acrosome already had this, okay, basically. And its enzymes help fertilization, help to break down the zona pellucida of the egg so that you can fertilize, well, the ovum, so you can fertilize it. It's not an egg until it's fertilized. So I need to be a little careful there because it is an ovum. Now, the neck is just the piece at the back here. The neck does hold some of the centrioles and some of the original nucleus of the first spermatid that was made. The cell, its original nuclei and things like that is kind of there. The centrioles, things like that. The mid piece holds all the mitochondrial spiral that's there to power the flagella for swimming, and the tail of the flagella. That's the only flagellum the human body actually produces. It's not the only flagellated cell in a human because bacteria have it, but this is the only one that is made from a human cell, and it swims. Okay. Now, sperm is produced. And when a sperm detaches, it enters the epididymis. And so it's made in the seminiferous tubules. It goes to the epididymis. It goes to the vas deferens. It goes in the ejaculatory duct through the urethra and out the penis. To help remember this, think 7 up. We start the seminiferous tubules to the epididymis, to the vas deferens, to the ejaculatory duct, to end for nothing because I want to spell 7 up. Urethra out the penis. Okay? That is the path of sperm to go out of the body. 7 up. Now, one of the last things we're going to talk about throughout our time here is how we control males' reproductive structures. So what I want to do is, as I start this, I'm actually going to draw it out kind of first and explain it a little bit with males. And what we're going to do is up in the brain, we're going to have our hypothalamus. Then we're going to have the anterior pituitary. Then we're going to have the testes. Now, we might know a little bit of this already. Let's say normally what's going to happen under normal thing, let's say that a male needs to produce sperm. What are we going to do? Sorry, I hit my microphone that's on my headset. We are going to make a hormone here called gonadotropin releasing hormone, GnRH. Now, there's going to be 2 impacts here, okay? First thing, the anterior pituitary release 2 hormones. One of these is going to be this hormone called FSH, and one of them is called LH. Now, LH and FSH have very different things. FSH job is to help produce sperm. Now, LH's job is to help produce testosterone. Now, kind of thing about it is that testosterone makes men like hairy apes. Testosterone, like hairy apes. LH is what helps that production, okay? Testosterone production is something like LH. Now, that will help sperm production, okay? Now, let's say the sperm levels have gotten high enough. What we're going to do is the nurse cells in the testes are going to make inhibin. So if this happens, we're going to have inhibin. One of these days, I will learn to spell. Inhibin will come back, and remember, its job is to negatively inhibit FSH release so that the sperm levels drop back down again, okay? And so that way, we can stop making sperm but still make testosterone, okay? Think about it that way. That's kind of what you want to do. Men we need testosterone throughout, even though we have enough sperm, we still need testosterone, okay? So I always like to say that hypothalamus makes gonadotropin-releasing hormone, GnRH, which actually causes the production and release of FSH and LH, luteinizing hormone. The FSH is sperm production. LH makes our testosterone because it makes men like hairy apes because we use androgens. So like hairy apes, LH helps us make our male androgen testosterone. Like hairy apes. Like hairy apes. Okay. Now, GnRH from the hypothalamus, it actually goes in the hypophyseal portal system to go down to the anterior pituitary. And it's released every hour to, 60, 70, 80, 90, hour, 2 hours, hour and a half, around there. So 60 to 90 minutes intervals. And this controls our production and secretion of LH and FSH. And testosterone gets released because we have LH. So testosterone every 60 to 90 minutes, so will be that testosterone surge as well. Now the FSH and testosterone, they target cells that produce sperm called the Sertoli cells. Okay, Sertoli cells. In the seminiferous tubules, you also have Sertoli cells, and they are going to FSH and testosterone, the nurse cells, called Sertoli cells. The nurse cells are targeted here. Remember, Sertoli cells or nurse cells also produce the ABP antigen binding protein to make sure we keep that up, and that will make sure testosterone levels are high to make sure spermiogenesis happens with antigen binding protein and FSH to stimulate sperm production. So sperm needs FSH, but it also needs testosterone. So ABP helps get the testosterone levels up to bind that up, okay? And then Müllerian inhibiting factor by the nurse cells or Sertoli cells. We'll talk about the Leydig cells in a minute. The Müllerian inhibiting factor that actually is also there causes regression of the Müllerian ducts, which would turn into the uterine tubes. And if you don't have that, you don't get rid of the ducts and the testes don't descend, called Müllerian inhibiting factor, MIF. Now, negative feedback mechanism, remember, if I have enough spermatogenesis, I have enough sperm. So if I need to produce it, we have GnRH, FSH, and inhibin are the main regulators of sperm production. Now, if we have enough sperm produced, inhibin will go turn off FSH production from the anterior pituitary. Now, it can also affect and suppress GnRH as well. And I'll kind of do that here as well. But we have this ability to do these negative feedback mechanisms, okay? I probably should have used red for that, but I forgot to talk about that. So if you have very high levels of inhibin and things like that, then you are not producing FSH. So if FSH goes up, inhibin goes up. Inhibin goes up, FSH drops. And then when FSH drops, we can then stop releasing inhibin and keep going back. When FSH decline, we'll stop producing inhibin and FSH goes back up. And what we do is we're constantly producing this and altering it, so we're always just producing appropriate levels of sperm cells. Okay, these are negative feedback. Elevated FSH will cause inhibin production, things like that. Okay, so it is that high level sperm because of FSH. Now, luteinizing hormone is produced by Leydig cells. Easy to remember. Luteinizing, who produces testosterone and the other androgens. Luteinizing is Leydig. Another thing is we can think of men who have a lot of testosterone might lie. Maybe. I don't know. So Leydig cells. LH affects Leydig cells. Both again with an L. Okay. That makes testosterone and your other androgens that are needed for appropriate sperm production. Now, testosterone helps spermatogenesis, especially maturation, spermiogenesis, but it also, testosterone affects male sex drive, called libido, which is in the central nervous system, which is all the related sexual drive behaviors that a man may do to try to get women in his sex drive. But it also affects the metabolism, like red blood cell, muscle growth, protein synthesis. Why men and their blood cell formation, why men who have low T will have low energy, so basically because they're not producing enough red blood cells, and also things like muscle growth, things like that, weakness, and things like that. They're also, and I've been reading some papers on it. It's come out the last year, papers from the last year about. There's also seems to be some good important things coming out about low testosterone and potentially things like depression, anxiety for males. Male mental health is another thing that's not taken very seriously. Testosterone helps produce male secondary characteristics. As I said, it makes men like hairy apes, where men grow their hair. Facial hair, for example, even though I don't grow a lot, a very thick, luxurious beard I don't grow, but I do grow it. And I have to shave the facial hair, the bigger body size and greater muscle mass usually of men, is also where men tend to deposit their body fat, like on their bellies. Also, the male reproductive glands are also maintained by testosterone. So, testosterone is not there. This can also potentially lead males to greater risks of reproductive cancers. As I'm beginning to realize. I need to do more research on that. Some papers I've been trying to read. I'm catching up on reading. I don't get to read as much primary scientific literature as I used to. I read a lot of primary scientific literature when I first started teaching, but I was an adjunct and I only taught a certain number of classes. I had more time on my hands and more free time as an adjunct faculty. And I was also still involved in research. Now it's kind of a little bit different, but I still try to read. So a lot of times I'll just read books that cite the literature, and then I get an idea of what literature I want to read better that way. But then if I don't ever read the primary stuff, I'm reading the people who are doing it or watching lectures associated to it. So anyway, that is it for this first part. I hope you find it useful. We're going to look at the female here in just a little bit. I will record that later on today. I've got to take a little break, though, and let my voice recover. But thank you guys so much. I hope you appreciate it. Once I'm done with this second section of reproductive, let me know on some things you really want to see. I've got some things I'm thinking about doing, some videos where I'm actually in them, plus some more topical videos like that I want to do and then I'm very interested in doing. So please let me know. Don't forget to give me a comment, like, or subscribe. But as always, thank you guys for watching, and I'll see you in the next one.